presentation. Sorry about this, everybody. We're having some sort of technical issue here. Our next presenter, as Wendy had mentioned, is Dr. Xavier Ortiz. Dr. Ortiz is an adjunct professor at IQS School of Engineering of Ramon Llull University in Barcelona. His research focuses on the development of targeted and non-targeted methods for the analysis of cyanotoxins and other pollutants in the environment. In the past, he worked at the Ontario Ministry of the Environment as a scientist, where he developed the method E3450 for the automated analysis of microcystins and Anatoxin-a using online SPE combined with LC QTOF-MS analysis. Welcome, Dr. Ortiz, and would you please begin your presentation?
Sure. Let me see if I can find this here. Can you see my presentation? Okay. Okay. So thank you, Ben, for the introduction, and thank you to all the previous speakers, especially Wendy Strangman, for this incredible introduction to the world of cyanobacteria. I'm going to start about talking about the cyanobacteria, and the last few years they have been getting a bad reputation, but really they're not that bad if you consider that they play a vital role in our ecosystem, because roughly they produce about 50% of the oxygen we have in the atmosphere. And throughout millions of years, they managed somehow to shape this atmosphere we live in with around 21% of oxygen. There's also been, they are used widely in the food industry as supplements. But it's true that when they grow out of control under certain conditions, they can be toxic, either by removing the oxygen dissolved in water and creating anoxic conditions where aquatic species suffocate, or they can produce toxic secondary metabolites that we know as cyanotoxins, as Wendy was introducing. There's been a raising awareness concern the last few years about algal blooms. These two figures kind of illustrate this point. In this first one, we can see the amount of times that algal blooms would be on the US media, whether it was news or the newspaper. And here we can see also an increasing number of publications focusing on cyanotoxins. And why do we see more and more often each summer, especially these algal blooms? Well, mainly combination of two factors: climate change, which increases the world temperatures and the water gets warmer, and human activities like agriculture, increasing the amount of nutrients in water. So this creates the perfect conditions for cyanobacteria to thrive in fresh water. As it was pointed out before, there's many different families of cyanotoxins. We can classify them into hepatotoxins, so those that affect the liver, or neurotoxins, those that affect the nervous system, the brain. And we noticed that the analysis is a little bit tricky, but compounds like microcystins, there's not only one particular microcystins, there can be hundreds of microcystins with little variations on their structure, which from a mass spec point of view, it totally changes the masses and the precursors and the fragments we're looking for. So this makes the analysis a little bit challenging for all these families. More from a European perspective, this is a review that it's a bit old, but it makes the point of what I'm trying to explain. Algal blooms have been detected pretty much everywhere that it's been sampled. Not only that, but most of the time, these algal blooms are toxic. This means that they had some kind of toxin in it, and probably this percentage is going to be higher because, as Wendy was explaining, there's more toxins than only the traditional ones that we're looking at. If we compare this data to other countries, we have similar results. There's been there's lots of evidence of algal blooms in North America, South America, Africa, Asia, etc., etc. What's interesting is that the percentage of toxic algal blooms remains about the same. So this means that we can pretty much see cyanobacteria anywhere in the world, and most of the time, these cyanobacterial algal blooms are toxic. From a drinking water regulation perspective, here in the European Union, we have a directive from 2020 that states that by 2026, all the member states should be able to analyze microcystin LR in tap water, and the concentration should be less than 1.0 micrograms per liter. And notice that we're just regulating one particular microcystin variant, and we know that there's other variants with similar toxicity. Also on small print, this directive also states that this parameter will be measured in the event of potential blooms in source water, meaning when the intake of the drinking water station looks kind of greenish, we're going to do the analysis. And this is a good proxy most of the time, but not always, because in this picture of a couple of samples they analyzed a while ago, this sample here looks fairly clean, and this one looks fairly dirty or with cyanobacteria in it. But when we analyzed it, we saw that really this one had no toxins in it, at least the toxins we were monitoring, and this one was loaded with toxins, in this case, anatoxins. So the visual inspection of the sample is sometimes yes, but not all the times the best proxy we can use. On the other hand, the World Health Organization is recommending maximum levels of 1.0 micrograms per liter of total microcystins. And most of the guidelines in North America seems to be shifting towards that direction. This is a more protective measurement, and it makes sense, but it poses an instrumental challenge, because how do we analyze microcystins that we don't have standards for? So here we're going to be focusing mainly on two toxins, anatoxins, with, as Wendy was explaining, they are very toxic. Somehow they've managed to fly under the radar until recently, when the World Health Organization recommended of not having anatoxin levels in tap water higher than 30 micrograms per liter. There's been many incidents in Canada and the US and worldwide of pets and dogs dying due to exposure of these toxins, and they can be analyzed by LCMS or bioassays like ELISA. And there's two main variants. There's Anatoxin-a, which we see in this picture here with a methyl here. There's homoanatoxin, which there's an ethyl group here. But Anatoxin-a seems to be the most common variant of this compound. We also have microcystins. They're produced by cyanobacteria. There's over 300 microcystin structures known to date, and they've been monitored for a long time in drinking water in North America. They can be analyzed both by bioassays or instrumental techniques like LCMS. And each country, or in the case of the US, each state within the country have slightly different guidelines. So it makes the comparison a little bit trickier when it comes to sharing data among states or countries. This is the generic structure of microcystin. This is microcystin LR. And notice that that is a cyclic heptapeptide with 7 amino acids. There can be many substitutions in each one of the positions, although most of the substitutions occur in position 2 and 4. So when I say microcystin LR, means that has leucine on position 2 and arginine R on position 4. And this amino acid ADA that is common in most of the microcystins, in all the microcystins with some variations. And it's a strange amino acid that we don't produce, and it's only produced by cyanobacteria, as we know so far. If we talk about numbers, and notice that this scale is in log scale, here in Europe, we only have one variant regulated, LR. We have roughly about 12 different standards commercially available, although there's over 300 reported variants. And if you take into account all the possible amino acid configurations that microcystin can have, so theoretically, that's the theoretical calculation, there can be over 200,000 different structures. Of course, there's a huge gap between the variants we know and the hypothetic variants. So this means that probably there's still a few microcystins that we haven't discovered. So in this study, we're going to focus on the internal standard. As you may all know, internal standards are important in analytical labs because they help improve the method's performance by increasing the accuracy and the precision and reducing the uncertainty. In the cases of methods that where sample preparation is really long, it can mitigate the effects of recovery law throughout the steps of extraction, recovery, etc. In our case, though, we're more interested on the how can it help the instrumental side. LC-MS is a great technique, but arguably its weakest point is electron ionization. We're not that efficient creating ions and putting them inside of the mass spec. And also we get this ion suppression when there's other compounds or matrix coming out of the column where our analytes may not be ionized as effectively as we would wish. So an internal standard should have a chemical behavior as close as possible to the analyte and not be naturally present in the analyzed sample. And in the case of mass spec, this means that when available, it's not always the case, mass-labeled internal standards are always the best option. Here we're going to be using, the goal is to develop a method based on LC-MS triple quadrupole for the analysis of 4 of the most common microcystins, LR, LA, YR, and RR, plus anatoxin in the same run. We want a method that is quick and high throughput, so we're going to do a large volume direct water injection to avoid manual steps like SPE. And we want a method that works for both simple, let's say, samples like drinking water and complex samples like surface waters or algal bloom waters. For this, we're going to be using CIL's nitrogen labeled internal standards for the 4 microcystins. So that would be the isotope dilution method. In the case of anatoxin, we don't have a labeled standard yet. So we're using deuterated phenylalanine, which chemically is close enough. Here we can see the structure of microcystin, the native compound, and the labeled compound. All the nitrogens are labeled with nitrogen 15, and this particular variant having 10 nitrogens. The mass shift is 10 m/z units. As for the LC-MS method, it's pretty straightforward. We reach the detection limits by relying on a very sensitive instrument, SciX 7500. And the chromatographic run is pretty simple. We use water with formic acid and ammonium fluoride to help ionization, gradient to acetonitrile at 0.4 mils a minute. And we're using this Phenomenex Luna Omega column, which is, it works very well for the retention of compounds that are small and very polar, like anatoxin. And we're injecting 250 microliters. We're not injecting the typical 5 to 10 microliter LC run sample. As for the ionization, we're working in positive mode. And the fragmentation, if we look at the MRMs, the precursor depends on the mass of the precursor, of course, but some of the transitions are common because some of them, they come from the same shared other amino acid, this fragment at 135 and 103. Here's the chromatogram of the mixture. As we expected, anatoxin being a very polar compound has shorter retention time. This compound has a secondary amine, and when there's formic acid in the solvent, it gets protonated, so it typically flies through the LC column. But in this case, we have a decent retention. And as for the microcystins, we can see like 4 different regions. We see compounds like RR that has 2 arginines. Arginine is a very polar amino acid. In the case of RR, we see lower retention times in reverse phase LC, but also we see the doubly charged ion because it's very polar. Compounds like LR and YR, which only have 1 arginine, we see a mixture of the singly and doubly charged adduct. So depending on the ionization conditions, you have to make sure that you're selecting the most abundant adduct, and they have intermediate retention times. And then we have other structures like LA, which not having arginine is not as polar, so higher retention times, and we only see the singly charged adduct. So although we would have to have as many label standards as possible, this selection of internal standards, it makes a lot of sense because they cover most of the retention times that we're going to see for the other variants. So if I wanted to analyze WR, which I don't have a standard for, probably the retention time is going to be fairly close to either YR or LR. Here, if we look at the chromatograms, the retention times, as expected, are virtually the same between the native compound and the labeled compound. And about the method, so we wanted something that it's high throughput. When comparing this to the traditional SPA approach, like the APA's 544 method, which is a very good method, it's bulletproof, but it takes a long time to do the manual SPA extraction, then evaporate the solvent, et cetera, et cetera. So what we do here is basically we take the sample, we freeze and thaw 3 times to lyse the cells and have access to the contents of the cell. And then we do the LCM-SMS analysis. By doing so, we increase significantly the lab capacity, the amount of samples we can run per day. We reduce the emergency response in case there's an emergency by maintaining the MDLs, and we need less people in the lab to prepare the samples. We wanted to evaluate the matrix effect when it comes to accuracy, because when we're having very complex algal blooms, we can get ion, most of the time ion suppression, sometimes ion enhancement, and then that can bias our accuracy. So we used this really complex sample that is actually a fountain that we have here at the university. We analyzed it during the summer, and there's actually cyanobacteria and toxins in this. And we tried 3 different quantification strategies. So external calibration, meaning no internal standard, using an internal standard like nodularing that it's a pentapeptide, and historically in many methods has been used in the past as internal standard for microcystins before the labeled compounds came out, and these nitrogen labeled compounds. And these studies were carried out at 3 different concentration levels, and each one by triplicate. So this is just a few chromatograms. Here on top, we can see the Q1 scan TIC, total ion chromatogram, and this gives us an idea of how the matrix that's coming out. So where there's a lot of the intensity of these humps is very high, we're going to get a lot of ion suppression because there's a lot. We don't know what this is, but there's some part of the matrix coming out. How is this going to affect our quantification? So, for instance, if I wanted to quantify microcystin LA by external calibration, I'm going to get ion suppression here. So my accuracy is going to be biased low. If I'm using nodularing as the internal standard, actually in this case, we're going to have more ion suppression on the internal standard than on the native compound. So the relative area is going to be higher than expected. So the accuracy is going to be biased high. In the event that we can use the native and labeled internal standard, the retention time is exactly the same. So accuracy should not be affected thanks to the matrix coming out. It can affect detection limits if really there's a lot of stuff coming out of the column and we don't ionize our compounds, but there's not much we can do about that. So I apologize for the amount of information in this slide. But as we can see in blue, for all the analytes at all the concentrations we tried, the best results we could get, the most accurate results were obtained using the labeled compounds. In some cases, the recoveries were pretty bad for LA, for instance, or here for RR. We could not quantify really RR with external calibration, not because we didn't see any peaks, but the peak that we saw was smaller than the intercept on the calibration curve. So actually that was a negative concentration, which obviously doesn't make any sense. But in all the cases, using labeled compounds can correct this ion suppression caused by the matrix effect, and we get fairly good results. So as for the validation, I'm running short on time. So we validated drinking water and algal bloom water For drinking water, it's a fairly more simple metrics. We had good recoveries and low RSDs, so we're fairly happy with that. As for algal bloom, the algal bloom sample, recoveries were also pretty good. Maybe for R still a little bit lower, but we're still acceptable. We're working on that. And the RSDs also acceptable. So we're happy with this validation data. Just to conclude, we developed this LCMS method for the analysis of 4 of the most common microcystins and Anatoxin-a in the same run, using the mass-labeled internal standards. And the method is based on a large volume injection to avoid SP concentration and saving time and money. The use of labeled internal standards significantly improved our accuracy because it mitigated to some extent the ion suppression caused by the matrix. And the selection of internal standards, the labeled standards, these commercially available through CIL, actually makes a lot of sense because they cover most of the microcystin chemistries in terms of polarity. So we can work with them. They work very well. And finally, the method that we validated for microcystins and Anatoxin shows good precision and accuracy in both drinking water and algal bloom samples. And thank you for your time. And I don't know, I'll be happy to take questions if there's any.
Okay, thank you very much, Xavi. We really appreciate all that information. Just a quick side note, we are already working on standards for all 200,000 of those variants that you're looking for. So we should have those probably in the next couple of years.
Excellent.
Actually, that's a joke, everybody. Please don't take me seriously. I had a quick question for you, actually. Now that you're starting to see in Europe regulations coming on board for levels of allowable limits, things like that, do you anticipate that there will be an EU-based standardized method upcoming, or is that in the works that you know of?
Well, right now we have this directive. Spain already has its own directive in terms of drinking water, but right now, for this directive from 2020, the goal is that by 2026, all the countries in the EU should be able to at least measure microcystin LR in drinking water. So far, we only have a regulation for drinking water, not recreational water. And of course, it's very limiting just having one particular variant regulated, because we know that there's many others, like LA, that we see all the time as well, and has a similar toxicity. But with time, maybe it seems that the trend is that all guidelines are shifting towards total microcystins more than specific variants. But that's a different question.
Okay, thank you. We do have a couple of other questions, but in the interest of time, if you could maybe answer those, Xavi, if you get a minute, the questions that came up in the Q&A. And sure. We'll go ahead and move on to the next speaker, if you don't mind doing that.